Looks like someone's hungry.
The phone is ringing.
I'm fasting. Okay, bye.
During starvation, our bodies undergo several mechanisms to generate ATP. The first one is carbohydrate metabolism. In the first 6 hours of starvation, store glycogen in our liver and muscle cell broken down into glucose to produce ATP. This process known as glycogenolysis. When our blood glucose level decreases, it will send signal to our bodies to secrete hormones such as glucagon and epinephrine. Glucagon produced by the pancreas while epinephrine is released by the adrenal gland. This hormone will stimulate the conversion of glycogen in the liver to glucose 1-phosphate. In this step, glycogen phosphorylase cleaves the alpha-1,4-glycosidic bond, releasing the glucose 1-phosphate from the glycogen chain. The next step, glucose 1-phosphate is converted to glucose 6-phosphate through the action of enzyme phosphoglucomutase. Glucose 6-phosphate is further converted to free glucose by the enzyme glucose 6-phosphatase and now ready to enter the bloodstream, raising the blood glucose level and provide available energy throughout the body.
During fasting, the person's blood glucose level is decreasing. Thus, it triggers pancreas to secrete hormone epinephrine into the liver for gluconeogenesis when the glycogen is depleted. Gluconeogenesis is the process of glucose synthesis from non-carb sources. So here is the process of gluconeogenesis from pyruvate. So the pyruvate here will be converted to oxaloacetate by the help of enzyme pyruvate carboxylase, and then it will be converted to phosphoenol pyruvate by PEP carboxykinase. Several steps from phosphoenol pyruvate will be undergo to produce glyceraldehyde 3-phosphate. The glyceraldehyde 3-phosphate then will be converted to dihydroxyacetone phosphate. The dihydroxyacetone phosphate will be processed to produce fructose 1,6-biphosphate, and then the fructose 1,6-biphosphate will undergo step to produce fructose 6-phosphate by help of fructose 1,6-biphosphatase, and then the fructose 6-phosphate will finally produce glucose, where the glucose is the main energy source for the synthesis of ATP.
Next, I will explain about the protein metabolism during starvation. Protein will undergo gluconeogenesis during starvation. So, what will happen to protein during gluconeogenesis? The protein in the skeletal muscle will hydrolyze into amino acids. The amino acids undergo transamination and deamination for the liberation of ammonia. Or on the other hand, the carbon skeleton of the amino acid is first converted to keto acids by transamination in order to produce glucose. Let's focus on the glucogenic amino acids first that are in the blue box. The alanine, cysteine, glycine, serine, threonine and tryptophan will converted to pyruvate or acetyl-CoA and enter the citric acid cycle. Next, arginine, glutamate, glutamine, histidine and proline will converted to alpha-ketoglutarate and enter the TCA cycle. Isoleucine, methionine and valine will converted to succinyl-CoA and enter the TCA cycle. Aspartate, phenylalanine and tyrosine will converted to fumarate and enter the TCA cycle. The last one, asparagine and aspartate will converted to oxaloacetate and then to phosphoenolpyruvate to produce glucose. Okay, now let's focus to the ketogenic amino acid. Isoleucine, leucine and tryptophan will converted to acetyl-CoA and enter the TCA cycle. Next, leucine, lysine, phenylalanine, tryptophan and tyrosine will converted to acetyl-CoA then to the acetyl-CoA before enter the TCA cycle.
So I want to tell you about the process of ketogenesis in general during starvation. Basically, during starvation, the body undergoes a series of metabolic changes to ensure the survival in the absence of sufficient food intake. Which then, one of the significant adaptation is the activation of ketogenesis, which allows the body to produce ketone bodies for energy when glucose level become critically low. So, here's how ketogenesis occurs during starvation. So first thing first is about glycogen depletion. So in this initial stage of fastings or starvations, the body relies on its glycogen stores. So the glycogen is stored in the form of glucose that's found in the liver and the muscles. So as a glycogen is used up, glucose availability decreases. Then we go to the transition to fatty acids metabolism. With the previous diminution of glucose levels, the body shifts to using fat as the primary energy source. As we use body fat, which is adipose tissue, is then being broken down into fatty acids and glycerol through a process that we call lipolysis. In general, the process of ketogenesis reaction occurs in the matrix of the mitochondria of the liver, which is hepatocytes. Thus, in this liver, these fatty acids are converted into ketone bodies. We will further continue to learn about the process of ketogenesis after these slides.
Lipid metabolism and gluconeogenesis during starvation are intimately related because the breakdown of lipid release substrates for glucose synthesis. Triglycerides, which are lipids stored in adipose tissue, are a vital source of energy when fasting. It have a 5-step, which is lipolysis, glycerol utilization, fatty acid utilization, acetyl-CoA and ketogenesis, and ketone body utilization. So, the first one is lipolysis. When a person is starving, their body breaks down fat by release more glucagon and other counter-regulatory hormones. The process of breaking down triglycerides into glycerol and free fatty acid in adipose tissue is known as lipolysis. For glycerol utilization, glycerol is released into the bloodstream as a byproduct of lipolysis. Glycerol is able to absorb by the liver, where it is transformed into glycerol 3-phosphate and subsequently into DHP, known as dihydroxyacetone phosphate. Through gluconeogenesis, DHAP can be further transformed into glucose and contribute to the production of glucose. For fatty acid utilization, fatty acids are produced through lipolysis and are a vital source of energy for many different tissues, including muscles and some organs. FFAs are delivered into the tissue, where the mitochondria use them to produce acetyl-CoA through a process known as beta-oxidation. Then we have acetyl-CoA and ketogenesis. It is not possible to use beta-oxidation produce acetyl-CoA directly in gluconeogenesis. Rather, it becomes a part of the Krebs cycle, which is the citric acid cycle, and aids in the synthesis of reducing equivalents such as NADH and ADH2. The liver uses extra acetyl-CoA that is not completely used in the citric acid cycle for ketogenesis. As a result, acetoacetate and beta-hydroxybutyrate, 2 types of ketone, are produced and released into the bloodstream. So the last step is ketone body utilization, which ketone bodies have the potential to function as a substitute fuel source, especially for the brain and other tissues. The brain eventually switches from using only glucose to using ketone bodies during chronic famine, which lessens the need for gluconeogenesis to produce glucose.